Hello, we'll be presenting on cranberries as a UTI preventative agent. So, over the course of this presentation, we'll discuss a little background, the methods used in our trial, the results, and we'll do a little conclusion and discussion. Now, let's start by discussing a little background. So, what exactly is a UTI and who does it affect? Urinary tract infections, UTIs, are a frequent ailment that have significantly plagued young, healthy women at a remarkably higher rate than men. Recurring UTIs oftentimes lead to heightened morbidity, discomfort, and healthcare costs. So, how are UTIs currently treated? Currently, antibiotics are the first line treatment utilized to remedy UTIs. However, with the continuous surge in antibiotic resistance, non antimicrobial preventative measures are necessary. So, why are cranberries commonly accepted as a UTI preventative agent? It is commonly speculated that cranberries possess non antimicrobial properties. Therefore, ingestion of cranberries is perceived by many to be a UTI preventative agent. To further support these beliefs, in vitro data has revealed that inhibitory activity cranberries have on the development of UTIs. The title of our trial is Recurrent Urinary Tract Infection and Urinary E. coli in Women Ingesting Cranberry Juice Daily, a randomized controlled trial. Amongst the authors, there are no conflicts of interest. The reasons they wanted to conduct this study is to investigate the validity of previous studies. There were multiple in vitro and also in vivo studies. Also, to the existing literature, this study will add data on cranberry's ability to reduce rates of UTI, asymptomatic bacteriuria, and urinary fimbriated E. coli in premenopausal women with a history of recurrent UTIs. The design of this trial was double-blinded, randomized, controlled study. And the primary objective was to compare the time to urinary tract infection and the rates of asymptomatic bacteriuria and urinary P. fimbriated E. coli during a 6 month period in women ingesting cranberries versus placebo juice daily. So now let's discuss a little review of literature that led our authors to begin this study. One review was Cranberries and the Chemical Composition. This study revealed that cranberries are abundant in nutritional components and many bioactive compounds that have antioxidant properties. Moreover, it revealed that the cranberries have a beneficial effect against urinary tract inflammation with both adults and children, cardiovascular disease, oncology disease, type 2 diabetes, and so on. An additional study assessed cranberry juice and how it lowered UTI episodes. This particular study was a randomized controlled trial, and the conclusion was that the consumption of cranberry juice beverage lowered the number of clinical UTI episodes in women with a recent history of UTIs. Lastly, another study I found was cranberries versus antibiotics for UTIs. This study was a non inferiority trial, so I wanted to see if one treatment was better than the other. In this one, it was revealed that Bactrim was in fact a better treatment and better preventative agent than cranberries. So in premenopausal women, consuming 480 milligrams once daily of Bactrim was more effective than cranberry capsules, which were about 500 milligrams twice daily to prevent recurrent UTIs. So now on to the methods portion. Who was included in this trial? Premenopausal women between ages of 18 and 45 years with a history of one clinician diagnosed UTI in the past 12 months. Also, the women had to agree to avoid all food containing cranberries or other vaccinium species other than a study product for the duration of the trial.
So, who was excluded? So, women with pre-diagnosed anatomical abnormalities of the urinary tract, women with known allergy or intolerance to cranberry products, women with symptoms of vaginitis or cystitis, women with asymptomatic bacteriuria, women with a history of diabetes mellitus, kidney stones, or malignant neoplasm, excluding skin diseases, pregnant or lactating women, women utilizing prophylactic antimicrobials or taking warfarin, and women who utilized antibiotics within the past 7 days or investigational drugs in the past 30 days. So, what was the experimental design? Recruitment methods employed for this study were advertisements in the local area and clinical referrals. Following written informed consent, the women provided a urine sample to assess for pregnancy and asymptomatic bacteria. Asymptomatic bacteria. If the urinalysis returned positive for either of the parameters, it signified that the individual was ineligible for the study. So the study participants were randomized into one of three groups in a 1:1:1 ratio, with a 4-ounce cranberry juice group, an 8-ounce cranberry juice group, and a placebo group. Randomization was stratified based upon site, and identification numbers were randomly pre-assigned for each study participant. At enrollment, each woman completed a questionnaire, a urinalysis and culture, and self-collected a vaginal and rectal swab. The study agents were provided by Ocean Spray Cranberries Incorporated, and the active juice consisted of 27% cranberry juice and sucralose. The placebo juice was similar in color and taste, however, did not contain any cranberry product. Women returned monthly for a follow-up for the duration of the trial, which was 6 months, and on any occasion if they experienced a symptom of UTI.
So, what were the outcome measures? The primary outcome measure was time to symptomatic UTI event, and it must have met the criteria for clinical or culture-confirmed UTI. The secondary outcome measure was the presence of functional P-fibrillation. The data categories collected included symptomatic UTI, culture-confirmed UTI, clinical UTI, and asymptomatic bacteria and P-fibrillation. Yes.
The statistical analysis, so the power analysis and sample size was determined by this. It was based on similar populations, and it was expected that 35% of women with a history of UTIs would have a symptomatic UTI reoccurrence within 6 months. So to achieve statistical power, it was determined that 350 women would have to enroll. 210 of these women would be placed into the cranberry group, with 105 in the 4-ounce group and 105 in the 8-ounce group, and then 105 in the placebo group. This would allow for a 10% dropout rate while still providing 80% power to detect an absolute reduction of 15% in the rate of symptomatic UTI reoccurrence. The statistical tests used were continuous variables were compared using the t-test, categorical variables were compared using the chi-squared test. The time to first UTI was assessed using the Kaplan-Meier method. The treatment arms were compared using the log rank test. Cox proportional hazards regression were adjusted for age, frequency of sexual activity, race, site, history of kidney infection, and number of bladder infections. All analyses were performed using SAS statistical software. And now on to enrollment and baseline characteristics. So 345 women were screened for enrollment. 136 were considered ineligible and 23 declined to enroll. In December of 2008, before the target sample size could be achieved, the study was terminated due to administrative and budget issues. 186 women, 100 from Seattle, and 86 from New Haven were enrolled and randomized. However, 10 individuals were lost prior to initial follow-up visit. The remaining 176 women were randomized into the cranberry group juice, 120, or the placebo group, 56. They were further randomized into subgroups. The 4-ounce cranberry juice had 63, the 8-ounce cranberry juice, 62, the 4-ounce placebo group juice, 31, and the 8-ounce placebo juice group, 30.
All right, so now to the baseline characteristics. Demographics and baseline characteristics were similar among all 176 women. The mean age was 25 years, 133 were white, and nearly all enrolled women were sexually active. The mean number of UTIs in the past year was 1.8 in the placebo group and 2.1 in the cranberry group. And all of these statistics may be found in the table that we have shown here. So now on to the results and statistical interpretation.
So the cumulative UTI rate was 0.29 in the cranberry juice group and 0.37 in the placebo group, with a p-value of 0.82. Adjusted hazard ratio for UTI in cranberry juice versus placebo group was 0.68 with a 95% confidence interval of 0.33 to 1.39 with a p-value of 0.29. The proportion of women with P-fimbriated urinary E. coli isolates during the intervention phase was 10 of 23 in the cranberry juice group and 8 of 10 or 80% in the placebo group with a p-value of 0.07. The mean dose adherence was 91.8% and 90.3% in the cranberry juice group and placebo group respectively. Minor adverse events were reported by 24.2% of those in the cranberry group and by 12.5% in the placebo group with a p-value of 0.07. So the statistical interpretations, a reduced hazard ratio of 0.68 for the incidence of UTI in women ingesting cranberry juice versus those ingesting placebo was noted, but was found to not be statistically significant. And adverse events were defined as, there were no serious adverse events that occurred in either group, but minor adverse events reported included constipation, heartburn, loose stool, vaginal dryness, vaginal itching, and migraines.
So now let's discuss the author's conclusion. So although the limitations of the trial hindered it from reaching full potential, the authors concluded that the study design was overall strong. The study results were portrayed in an unbiased manner. However, throughout the results, it was emphasized that cranberries may be a UTI preventative agent when compared to the placebo due to the hazard ratio of 0.68, although that is statistically insignificant. The cranberry juice did not significantly reduce UTI risk compared to the placebo. A reduction of urinary P-fimbriated E. coli strains in the cranberry juice group compared to the placebo group was noted, but was also found to be statistically insignificant. The author suggests that a larger sample size is required to prove statistical significance. Moreover, the cranberry juice group experienced reduced infection with P-fimbriated E. coli strains, as I previously stated. Consequently, the authors suggested an additional observation, specifically a cohort clinical study, be pursued in patients infected with P-fimbriated E. coli. So now, after this, we're going to discuss what we think. We personally believe that women of all ages suffer from chronic recurring UTIs and believe cranberries may be a preventative agent. Therefore, a strength of this study is its clinical relevancy. Additionally, the women randomized to each group were very similar at baseline, which lessened the likelihood of selection bias. Both the cranberry and placebo products were well characterized and standardized, and the deployment of multi-center enrollment allowed for the study to be reflective of women in different areas of the United States.
So the small sample size did not allow the authors to achieve statistical power. Therefore, this study was unable to detect an absolute reduction of 15% in the rate of symptomatic UTI recurrence. So an additional weakness of this study is adherence was self-reported instead of determined by urine metabolites. Individuals may have been falsifying their adherence or were unable to recall adherence. Consequently, this study may have been susceptible to recall bias. An additional weakness we identified is the lack of diversity in the study groups. A large percentage of the women enrolled were white, which may not be reflective of the entire population.
And moreover, the taste of cranberry juice may not be appealing to all individuals. Therefore, many individuals may have not been adequately abiding by the trial's protocol. And in future studies, we believe that compounding the cranberries into a tablet, a capsule, or even maybe a soft gel may prove to be more beneficial.
So in closing, we agree with the conclusion that cranberry supplementation does not reduce the rate of recurrent UTIs. Based on the presented statistical analysis, Cranberries do not reduce one's probability of UTI reoccurrence. Consequently, we do not find it medically appropriate to further encourage patients to ingest cranberry juice or supplements to prevent UTIs.
And these are the references we utilize to do our journal club. If y'all have any questions or like to give us any feedback, feel free to leave it down in the comment section below. This concludes our presentation.